Thank you, Monica. Based in part on presentations like yesterday's by Levi, I've modified what I'm going to talk about today to focus a little bit on one aspect of how we partner to try to accelerate the movement of knowledge into clinical practice. And, you know, if you sort of think about how you partner, there's another piece to that, which is what do you have to be an effective partner? How can you organize your institution or your programs to be more effective at partnering and creating the interfaces where partnering will happen? I think everybody, you know, has been talking about how this process is not a straightforward linear process. It's one that's iterative, where the points of interaction that can accelerate translation can occur at different spots in the process, and that you have to have those interfaces in place. So I'm going to talk a little bit about what we've tried to do at Dana-Farber and in our partnerships with the Brigham and Children's and Mass General, Beth Israel, the Medical School, School of Public Health, our Dana-Farber Harvard Cancer Center. A lot of this is intra-institutional because those are the resources over which I have the most direct control, and not offer this as either a model or a best practice or anything. I don't think it's either of those, but just simply one example of how we try to organize ourselves in order to be more effective as a translational engine and a translational partner. So I'm going to step back to what the Dana-Farber was like after it had gone a major reorganization right around the late 1990s when I arrived in the year 2000. All our faculty, by the way, are Harvard Medical School or Harvard School of Public Health full-time faculty. We have a few who have appointments primarily in one of the colleges in Cambridge. But we're organized into 8 internal departments, and as you can see, 5 of them are kind of like you'd think of a Department of Medicine or a Department of Pediatrics, a lot of physician scientists, clinicians, some people who are more into education, but sort of the multi-mission, and then 3 that are basic science departments. And for the size of our overall faculty, these are relatively large, and they maintain a strong interface for us with what we call the quadrangle at Harvard, the actual place where the medical school, the preclinical departments like cell biology, et cetera, are. One thing, though, about these departments, in contrast to a lot of medical schools, is that these are not terribly powerful as major units of resources. They're the academic homes for all of our faculty, but not the major place where resources are centered. Those would be our disease programs and disease centers, actually developed under Lee Nadler's leadership in the late 1990s. So these look a lot like what I think would be familiar to any cancer center right now. They tend to be anatomically based. We have a GYN center, a breast center, a GI center, et cetera. And then our early drug development center, where we do a lot of our phase 1 work. They're the sites where the clinical trial activity goes. And I would say that even by the time I got there, it was already becoming apparent that these were great at dealing with the fact that oncology care is becoming increasingly subspecialized. They were great in being interdisciplinary, multidisciplinary sites: surgery, radiation oncology, nursing, pharmacology, medical oncology, and so forth, came together in these centers. But like I think I've learned after 25 years now in academic leadership roles, that the natural destination of any program, institution, initiative that you create is to become a silo. And that these were becoming somewhat siloed around their particular cancers that they were interested in. And in fact, as I'm sure many of you are experiencing, these are becoming subspecialized and developing programs within themselves. It's no longer just GU oncology. There's renal and bladder and prostate and so forth. Our clinical trials happen there. I mean, that's the site where the activity is. But they're organized through this consortium cancer center that we've talked about in many places before. I just mentioned the members of it, where we have a single IRB that oversees trials in all the locations. So that was the setup, and we realized we started to do a strategic planning process around 2002 that we really needed to amend the model, that the pace of putting translation into clinical impact was way too slow, that while these serve many good purposes, primarily around the interface with patients, clinical and clinical trials, that they weren't a great way to try to move science into oncology practice. And through a strategic planning process, we decided we needed two things. We needed centers. We needed centers focused around the science, and those centers needed to be structured in a way that fostered translation, and that in contrast to a lot of research centers where the end product would be Nature papers, or the end product and funding and sort of organization and oversight of the centers would be very much like happens in academic departments, sort of academic metrics, that these would have much more of a project management idea, that there would be goals that these centers would achieve in terms of bringing things closer or actually into clinical practice. We created 12 of these out of that thing, and built our whole capital campaign around these. And the ones that are in bold here are just the ones I'm going to mention. You see, some of these are no-brainers. We had to organize, and Levi talked beautifully about the work that's come from the Genome Discovery Center yesterday. Some are a little bit flyers, like a Center for Cancer Systems Biology, that we think someday we'll be able to use these kinds of tools in a more translational way that's still, kind of out there. Some are built around particular modalities or just the kinds of capabilities you need. But these, the ones that I'm going to talk about, are the ones that we thought would sort of create a translational interface that would allow us to partner more effectively with other academic institutions, biotech, pharma, et cetera. And the organizational principles of these, forgetting what the content is for the moment, was that they be founded on business rules and accountability. So that, we would have to develop a business plan that led to self-sufficiency with 5 years, that the purpose of the business plan would be to create ways that people could bring projects, very goal-oriented projects, into these centers, and whatever it is the center brought to the table, mouse modeling or xenografts or molecule, lead molecule development, whatever it was, that there would be, expectation that the work that was coming out of these centers would be leading to something that would move something closer to a clinical application. The other thing that we did, and this takes advantage a little bit of the fact that the hospitals in Harvard are independent institutions corporately. I mean, we all have Harvard faculty appointments, but we can hire people within our own employment system. As we created a hiring track with 6 levels of professional advancement for research scientists. These folks can have Harvard faculty appointments. They're never, because they tend not to be working in the usual academic faculty model, going to rise much above the level of assistant professor in the Harvard system, but they can rise to very senior and highly influential levels within the Dana-Farber, and they populate these centers with people frequently recruited from industry who have more of that project-oriented focus. And all of this stuff notwithstanding, perhaps the most important part of creating these centers was creating a place in the institution where scientists with this kind of background and orientation had a home and could make a contribution and could have impact and could have upward mobility professionally. And we created the Office of Chief Scientific Officer. Barrett Rollins has filled that office since we started it, who oversees all this with the help of faculty and external advisors. So that was the idea of the center. We also realized that we had capabilities that were scattered and needed to be organized into programs. One was in chemical biology to generate new compounds that could potentially be drugs or at least tool compounds. And so we started that program. We've got 4 senior chemists now on the faculty who are doing that. And finally, a place that you can think of as a sort of mouse hospital that we raised some funds for. This is down in our south campus, or the dry dock campus. We're able to get space quite cheaply so that we could put a fair amount of space there, where we have mice that you can handle behind the barrier. These are mice that can be engineered or manipulated however you want, and go through a series of imaging steps as well as treating them and monitoring their response. And this allows us to do this approach of co-clinical trials. So with that, the key to making this work are a couple of centers. One is the Belfer Center for Applied Cancer Science. Ron DePino and Linda Chin started these. Kwok Wong and Pasiani now lead these. And you can think of this as a place where whether targets are generated by faculty or a company, Merck and Sanofi have been 2 that we've worked with very closely on this, have targets that they'd like to see were validated. They can take them to the Belfer, and there's a series of capabilities in there, largely around mouse modeling, but xenografts, genetically engineered mice, and so forth. And partnered with this, and it really shouldn't be an arrow because we basically see these as overlapping centers, is a place where you can interrogate molecules. And as you can imagine, the arrows are linear here. There's a lot of back and forth interaction. And once again, it's to create this interface where faculty, things coming out of our program in chemical biology, or any kind of external partner, you could bring a target, you could bring a molecule, and you begin to see that the idea is to create this iterative interface where you can look at these in mouse models, look at these in cell models, look at them dynamically with imaging, have them monitored through our Center for Oncologic Pathology. All the things that we talk about you need to do to decide if something is eventually going to end up in an early phase clinical trial. So the Belfer, in the way that it's organized, is really at the nexus of all this, because it is very much organized around milestone-driven structures. Their academic approaches are taken, but the idea is to keep moving these things through and decide at an early stage whether they have a future or not as a target or as a molecule. And there are about 60 staff scientists employed in this now who are experts in bringing whatever it is, the modeling, the analysis, to bear on the program. And then when you identify these targets, you can take them to the next step. So there's a movement back and forth, in and out of the Belfer, back into faculty labs or back out into the discovery engine or the application engine of the external partner. And this so far seems to be working very well. Initially, we worked closely on a longitudinal basis with, I mentioned Merck and Sanofi, with just a couple of pharmaceutical companies and a select group of faculty focused on a small number of cancers. But now we've broadened this and are bringing in more clinical trials and new pharmaceutical partners to this. So the idea is to create this to have a broad interface where people who see something as an opportunity for translation can actually test it and actually see whether it is going to move to the next step by actually trying to move it to the next step. And this is just in the lung cancer program, Posse and Kwok's work, of some of the things that have come out of work that has used this model of these integrative centers, used it to develop this idea of co-clinical trials, where you start a trial in a mouse model at the same time you start a late phase 1 or phase 2 trial. in humans. And we've had at least one example where we think that has helped to accelerate and make the trial in humans smarter. And you see that these are just about 8 months apart in their publication, the animal and the next phase human trial. Then one other thing I'll mention in closing is a longer standing partnership that we've developed over the years with Novartis, which is got a little bit more of a traditional base. And that is that Novartis provides funding in the form of grants to which investigators submit applications on an RFA basis. They're reviewed by a mix of Dana-Farber and Novartis scientists, and these are applied science grants. They're high quality, I think, in terms of the science that's actually done under them. But again, they're focused on particular targets or particular molecules that are of interest to Novartis. And the people get 2-year grants, and they have freedom to publish and all the kinds of things you'd think about with intellectual property. But a result of that has been, if you look at Novartis's oncology portfolio, that a majority in one way or another, and we haven't been the leaders on many of these by any means, but things that have moved ahead through the pipeline, and we like to think a little faster, that one way or another our investigators have done something to move these to the next phase closer to actual registration. So I think underneath whatever system it is that an institution decides to do to solve this problem, there are some common things we've learned from our experience. One is you need a great group of investigators, and they have to be a mix of basic, clinical, and translational. They have to be people, we're lucky to have these for the most part, who want to collaborate and interact, have their core scientific interests, but a part of their effort they want to do in this more team or goal-oriented, applied kind of scientific application. You need a place where there's the expertise in understanding what the clinical problems are. That's the disease center leadership that those disease centers have to have embedded in them. Some of these investigators, they can't be purely clinical centers. They have to have many of the features of a strong academic department. And then we think the integrated research centers is what creates the broad planes of interface where these things can happen. And of course, as we did with some of our centers, you need the technological background that we talked about through many meetings, including yesterday. And then that this broad interface allows multiple points of entry and exit and re-entry for this process of partnering and translation outside the immediate academic sphere. So I think the other lesson we've learned from this is that the usual model of how you share the potential results of this can change with this in a way that, you know, the usual way we'd set up these intellectual property agreements is you make a bet that you're gonna get My Fair Lady instead of the play that never makes it to Broadway. And so everybody protects jealously that potential big hit. But if you actually do sort of a net present value analysis, that getting upfront research support for these in terms, in return for licensing terms that are friendlier to the pharmaceutical partner is actually a better deal. You still, if the big hit happens, you still have a potential reward down the pike. But the value of the research support and the chance you have to make an impact with present day support for a discount down the road in the IP arrangement, we think is a much wiser way to approach it, and has been part of allowing us, I think, to have better relationships with the pharmaceutical industry. So with that, I'll stop and thank you for your attention.
Thank you, and we look forward to the panel discussion where we can talk about how this all fits together. Next, I'd like to introduce Dr. Renaud Capdeville, who is the Global Head and Vice President of Oncology Global Development at Novartis.